So for the second talk in this session, we have Professor Ines Thiele. She is the principal investigator of the Molecular Systems Physiology Group at the National University of Ireland, Galway. Her research aims to improve understanding of how diet influences human health. Ines has been pioneering models and methods allowing large-scale computational modeling of the human gut microbiome and its metabolic effect on human metabolism. And Christian showed the Agora models yesterday during the course. Those models were developed in Ines's group. So her group has been doing phenomenal work in this field. Just to give a brief background of Ines's academic profile, she earned her PhD in bioinformatics from the University of California, San Diego in 2009. From 2009 until 2013, she was an assistant professor at the University of Iceland. Later, she joined as an associate professor at the University of Luxembourg. And since Feb 2019, Ines is a professor for systems biomedicine at the National University of Ireland, Galway. She is the recipient of several awards like the Attract Fellowship, Embo Young Investigator Award, and she has also got the prestigious ERC Starting Grant. She's an author of more than 90 international papers, so she has done amazing work, and reviewers for multiple journals and funding agency. She's particularly interested in applying her computational modeling approach for better understanding of inherited and neurodegenerative diseases. Without further delay, Ines, I would like to welcome you to speak on in the symposium.
Thank you very much, Priyanka, for the extensive introduction. So that means I don't have to introduce the purpose of the lab anymore that detailed. Jason, good to hear you, and it's all right for the 5 minutes or 2 minutes, whatever it was. It's a great pleasure to talk to all of you and to introduce our work, particularly the most recent work we've been doing in the laboratory at the National University of Ireland, Galway. And as Priyanka said, the focus of the lab is really to understand how diet influences human health. And when you talk about diet, you really talk about the microbes that live in us and around us, and more specifically also about the gut microbes. The gut microbes are of particular importance, and you surely know about that, because they have many beneficial aspects for us beyond short-chain fatty acids. They're producing precursors for neurotransmitters. They're producing neurotransmitters. They're producing vital amino acids. They help us to digest food ingredients that we cannot digest otherwise ourselves. And so when we got started with this work about 10 years ago, we really kind of realized we have to go to the gut microbiome. Now, as Jason nicely introduced the metabolic reconstruction approach, I will only glance over that. But traditionally, what has been done for the metabolic reconstructions is that you would take, obviously, the genome annotations, which and list all the metabolic enzymes and transports known to be encoded in a target organism that you want to reconstruct, as well as you perform an extensive literature review. And that meant that the PhD student spent many hours reading papers about the biochemical characterization of single enzymes for hundreds or even thousands of enzymes, and put that information together with the genome annotation in a structured format into the computer. Now, on the right-hand side here, you can see that what we collect at the end in the computer is really a knowledge base, because it captures what's known about the target organism in terms of metabolism and physiology based on literature. So really the literature review is crucial here. And hence this approach has been also called the bottom-up approach. But at the same time, the way it's been encoded, through the stoichiometric matrix that Jason introduced, it is also a computational model. And by using constraint-based reconstruction and flex balance analysis, we can actually very efficiently and quickly compute metabolic traits of these in silico organisms. And more particularly, it allows us to go large scale quite easily. And for many of these metabolic reconstructions, but by far not all, there exist also metabolic maps which help you in visualizing the content of the knowledge base and the computational models. So basically, these reconstructions serve as a blueprint for consequent condition-specific metabolic models. So one reconstruction can give rise to many different models which are constrained on experimental data, omics data, just as Jason showed you, but at the heart of it lies the metabolic reconstruction. And that integrates the biochemical, genomic, and physiological knowledge for the target organism. Now, here's shown the map for human metabolism, but there are for many microbes, hand-drawn maps as well available. And so this visualizes you the content of the knowledge base. Each node here is a metabolite and each link is a biochemical transformation or a transport reaction. And so this wiring diagram is quite similar to a map of a city, if you wish, or a tubing map. And so the way the computations work here is that we can take a particular enzyme or gene product, map onto it experimental data, transcriptomic data, just as Jason mentioned, and tune how much flow or flux goes through every single of these links. And that's very similar to the pipe system or tube system shown here by opening or closing the valves. And with that, you change the properties of the metabolic model. And the beauty on the approach is that you can not only predict locally the consequences of such a change, but globally throughout the network. And this is, of course, also the case if you have different media composition or different dietary composition. Think about a high-fat diet versus a high fiber diet. So you have different inputs. So you change the valves here, and that triggers throughout the network and enables you to produce different biomass yields or energy yields. Now, if you want to go to the gut microbiome, which is very personal, time-dependent, space-dependent, and different between the individuals, we're talking about hundreds of thousands of different strains and species. So how do you accelerate this reconstruction approach that I showed you earlier? And that was a challenge we tackled a few years ago by creating a pipeline that is data-driven, of course, driven by the genome annotation, but also experimental data derived from the literature. And the pipeline that we developed makes great use out of the K-base, out of the Argonne National Laboratory, and also the Model Seed resource, which provides us with high-quality draft reconstructions. We found that particularly for gut microbes, which have less experimental data available and which might be phylogenetically quite distant to model organisms, it is very important to have very good genome annotation. So we spend a lot of time and effort in the group to do comparative genomics using majorly PubSeq and refine the pathways and the gene annotations across the microbes that we are reconstructing. And then, of course, we use as much as possible biochemical data. We have an in-house database, and I will show you the link later on, which uses its own nomenclature, and for convenience, we translate the draft reconstructions into this in-house nomenclature. If there are novel pathways described for organisms in the literature, we tend to add those ones on a regular basis, and that will require us to extend the pipeline and make revisions to the pipeline. Overall, the pipeline looks like this. We take the draft reconstructions from either K-base or modest seed. We convert it into our own nomenclature. We check for thermodynamic consistency. We apply a Western diet, which is basically a rich medium diet, to perform gap filling. And then we use information that we gathered from the comparative genomics to also further refine the reconstructions, correct the draft reconstructions if necessary, and also fill the gaps based on experimental data. And that is by now done largely automatically, but it does require to make certain adjustments and interventions manually and to inspect certain inconsistencies manually. So it's not a fully automated process, and to the best of our knowledge, it probably never will be, because there are always cases which are ambiguous and require more experimental data and additional review of literature. And then we have developed a test suite for quality control, both in terms of the ability of the reconstructed microbes to produce certain fermentation products, byproducts, or also grow on a defined media and carbon sources. And if the microbial reconstruction passes the test, we call it a semi-curated, semi-automatically curated reconstruction. If it doesn't pass it, it will go through another iteration. So by using such a semi-automated pipeline, we were able to largely accelerate the reconstruction process and generate, at the time it was 773 microbial reconstructions, which covered 14 different phyla. Now we have publicly available 818 of these microbes. And to our, well, those ones are accessible via the Human Metabolic Network database that I mentioned beforehand, and the link is given here. So you can pass it, or you can download the content, you can download the reconstructions, you can access the database via an API, or you can query its content via the website itself. Importantly, it also contains information about nutrition as well as human metabolism and disease. For us, quite exciting is there have been a growing number of papers using our resource in many different ways that we ever been able to think of, and that's quite exciting and rewarding, because we do this work for the community and we share this work openly with the community. So it's great to see that researchers take on to that. Now, for our own interest, we got quite intrigued about, really when we started working on data for Parkinson's disease, is that many of the elderly people that are affected with Parkinson's disease do take additional drugs to the main treatment, which is levodopa. And so really the challenge for us became that how to account for drug metabolism. In particular, there's increasing evidence that gut microbes can metabolize in one way or another on a large scale drugs that we take regularly. And so, so far, there are many reviews now available that describe in general the microbial activity, which can be either direct or indirect. And of course, drug delivery systems and pharmaceutical industry has been making use of these properties since many years by, for example, giving a prodrug rather than an active drug, which is then converted into a prodrug or an active drug by the gut microbiota. But also the microbiota can inactivate a drug or create toxic metabolites, which then might be in part at least causing the adverse effect that people are reporting. Then there's of course the antimicrobial and selective microbial properties of those drugs as well. But the indirect mechanism that also involves metabolism is that the gut microbes can produce certain metabolites such as paracresol that then interfere with host drug metabolism directly. So we set out to review the literature that's there about drug metabolizing enzymes and trying to represent them computationally. And so we found that to date, there are at least 15 known drug metabolizing enzymes, which are encoded by 25 microbial genes. Here's a table given. I'm just giving you the enzyme classes, the reaction types, and also the effect of the activation. Now, the important part is that many of this information has been generated using model organism again, and there is very limited knowledge available on a large scale of which microbes possess which enzyme and has which activity. And there are very recently papers coming out now to describe on the microbiome level the drug metabolism, but there are very few papers and still they do not characterize on the large scale per microbe the drug metabolizing capabilities. So using our comparative genomics approach, we set out for these 25 drug metabolizing genes to query over 5,000 genomes, gut microbial genomes, for the presence and absence of these 25 genes, thereby trying to get a strain level characterization of drug metabolizing capabilities. And we integrated those ones then with the microbial reconstructions. So we extended our first compendium that contained about 800 microbial reconstructions to now more than 7,000 microbial reconstructions, but we really focused our work on drug metabolism. And so we capture now these 25 drug metabolizing genes corresponding to 15 enzymes, the subcellular localization, which been predicted, and also we predicted 12 drug transporter genes. And that has been done across over 5,000 of these 7,000 genomes. And showing you the table from earlier on, again, we can now ask the question, well, what is the distribution of these different enzymes in the microbial strains, and which are the highest abundance ones? And what you can see is that certain central metabolic enzymes like the cytidine deaminase, which has of course other roles in metabolism than drug metabolism, is present in 80% of the microbes, while certain other enzymes like the dopamine dehydrogenase is only present in 3 of the analyzed genomes, consistent with other data that is out there. To really describe the metabolism of the drugs and enable actually drug-specific microbial metabolism, we also went through the literature and described or identified the mechanism by which the microbes metabolize the drugs and the different drug derivatives. So we have done that for almost 100 different prescription drugs, providing now a very comprehensive database across many different drugs for many different gut microbes in their capability to metabolize those drugs. Here's an overview given in form of a circus plot that shows you which phyla can metabolize predominantly, or which contains predominantly which enzymes. And as expected and mentioned earlier on, the cytidine deaminase is found in 12 of the phyla, the nitroreductase in 13, and then there are ecatellalinter specific drug metabolizing enzymes which is consistent with the literature. Not so surprising, probably, the species with most drug metabolic capabilities were Escherichia coli species. There's a very, Escherichia coli are very versatile, so that's not very surprising that they have many of these capabilities. We then went into the literature and identified information and collected information about microbes, either strains or species, and the ability to metabolize certain drugs. This information has not been used during the reconstruction or the comparative and genomics exercise. And so we got 238 drug microbe pairs. And the question here was then, if you take the metabolic models which contain drug metabolism for each of the microbes, how often do we predict correctly or correctly that they can indeed metabolize these the given drug? And you can find that we have almost 80% accuracy and a very reasonable sensitivity and specificity. So importantly, this is an independent data set. The Reconstructions never seen these data. So I think that's quite motivating that indeed these reconstructions represent for drug metabolism what might be happening in the given organism. Now with this a large set of microbial reconstructions at hand, we can now actually go further and ask the question, how do the drug metabolizing capabilities differ between individual microbes? And to address this question, we took a very comprehensive metagenomic data set from a Japanese cohort, actually, of 365 colorectal cancer patients and 251 healthy controls. Yoshida et al. Published that just last year. And we used our microbiome modeling toolbox, which is part of the COBA toolbox, to map the provided species abundance data for each of the samples onto the Accor 2 reconstructions. The data were only provided on the level of species and not on strains, even though the Accor reconstructions are on the strain level. So we generated, using the microbiome modeling toolbox, pan-species models. And in fact, we could actually map 97% of the named species, as opposed to some 70% if we were to use Accor 1 only. Now, we created a microbiome level model for each individual. So for each of these over 600 individuals, we have one model that represents the metabolism of each of the microbes that have been identified within the samples. We then subjected each of these microbiome models to an average Japanese diet that we derived from literature, and we added individually the different drug metabolites. And we asked, can we find or predict, using flex balance analysis, an individual-specific drug metabolizing potential, and does it for a given drug differ between individuals? And so here I'm giving you an overview of the drug metabolizing capabilities across these 600 samples and individuals. And you can see in blue where there is a conversion, just presence and absence, or qualitatively, if you want, in the microbes. And this is the case for most of the drugs. And in red, there's no conversion possible. And in fact, you can find the basilacide drug, which is the active form, the 5-amino-salicylic acid, and the inactive form is the Ines Thiele version of this drug that only a subset of the samples that we analyzed can actually carry out the metabolism. And in fact, it's 42% of these microbes can do the metabolism qualitatively. Quantitatively, all of these microbes microbiomes show difference. Now, since those ones are mechanistic models, which basically have the metabolism of each species within a microbiome, we can actually go back to the models and ask the question, well, where are the bottlenecks? Which species are the bottlenecks for drug conversion capability? And actually which reactions are causative for that as well. And I'm showing you here the conversion of a bisalicide, the activation from the prodrug into the active drug. And you can see that you had to have certain species in the microbiome in order to do the activation of the drug. And similarly, there are bottlenecks in the inactivation of the drug. And so this is one of the great advantages of the mechanistic model that permits us really to dig down into the pathway level and reaction level to identify what is happening and where are bottlenecks. Another example is levodopa metabolism and a digoxin metabolism. Both pathways are specific within the set of 7,000 microbes for Ecotella species, and similarly, those ones provide the bottleneck. The first step, the conversion of levodopa into dopamine, is done by Enterococcus faecalis. And you can see here that actually there are differences between the individuals. So even though you have the enzyme present, not in all cases, you get a one-to-one secretion flux, predicted secretion flux for tyramine if you give levodopa with the diet. And similarly, this is not a one-to-one translation in the conversion of dopamine to tyramine down here, and that is to do with the presence of Enterococcus and Ecotella and their relative abundance as well as precursors. And again, you can go down to the pathway level and really investigate what's happening, what's missing, what could be substituted. You could start thinking about products and probiotics and prebiotics here to activate certain metabolic pathways present in the microbiome, or to substitute the lack of certain microbial pathways. We can also then, given the clinical data that were provided by the cohort, ask the question whether the drug metabolizing capacities vary, for example, with age or BMI or disease state. But we did not find any strong correlation with the disease state. We did find changes over the course of age for the ability to inactivate 5-aminosalic acid. In fact, this ability increased with age. And similarly, there was a dependency of gemcitabine with BMI, whereby it increases the secretion potential with increasing BMI. So in summary, I presented you now a full-scale assessment of the drug metabolizing capabilities of the human gut microbes, which we done based on literature review as well as extensive comparative genomics. We have now a first time a large-scale description of strain level capabilities of drug metabolism. We have defined the reactions carried out by the gut microbes for almost 100 different drugs, so that you can actually investigate the role of the different microbes, either individually or on the microbiome level, for these different drugs, individually or in combination. I showed you that we could use microbiome level predictions to distinguish quantitatively as well as qualitatively differences between individuals. And there it becomes very interesting to now starting to think about responders and non-responders who might develop adverse reaction to a particular drug and not, and what could be potentially a microbial cause for that, and how could one prevent that by either using an alternative drug or by administering probiotics and prebiotics. I demonstrated to you that we could identify limiting factors or limiting microbes and pathways in the different microbiomes, so bottlenecks, as well as correlate age and BMI, but we found also gender and disease stage. We also investigated different diets in addition to the Japanese diets, and found that at least for some of the drugs, it's predicted to make a difference. Now, all that work would not have been possible with a lot of people involved. The driving person here being Almut Heinken, which is in my group, and she's been really the head of the microbiome work and driving the Agora 2. Dima Rafshi, which been driving the comparative genomics work, and many others have been involved over the years. I would like to help highlight that we have open position in the laboratory on all different levels, and I'd like to thank you for your attention, the organizers for giving me the opportunity to present our work, and yeah, I'm happy to take questions. Thank you very much.
Thank you, Ines, for such an amazing talk. And it was good to know like so much work has been done in your group for the microbiome analysis.